Welcome, deep divers. Today, we're tackling one of life's really big paradoxes, the oxygen that fuels you, literally right now as you breathe. Well, it's also a bit of a threat. It can turn against your own cells. It's this incredibly delicate balance, and that's what we're diving into today. We're digging into a fantastic chapter from Marks Basic Medical Biochemistry to really explore this, double-edged sword of oxygen, our mission. To figure out how oxygen keeps us going, but also how it can go rogue, what causes the damage, and importantly, the amazing defenses your body uses. Think of this as your shortcut to understanding this constant biochemical battle inside you.
It really is fundamental, isn't it? Both in biochemistry and when you look at clinical situations. What's so fascinating, I think, is how something so absolutely vital can, with just like a small change, cause so much cellular trouble and lead to disease. We'll follow this thread from just, everyday cell stuff all the way to big conditions, things like Parkinson's, heart damage, even some cancers. Understanding this paradox is really key.
Okay, let's start with the basics. Oxygen's essential role. We need it, obviously. It's crucial for making ATP, the cell's energy money, but also for detoxification, building things. In all these key jobs, O2 is there, ready to accept electrons, doing critical work. But here's the twist, right? When oxygen accepts single electrons, not the pairs it usually handles, that's when it changes. It becomes super aggressive, highly reactive. These are the oxygen radicals we hear about, like a molecule suddenly going rogue.
Exactly. And a radical, fundamentally, is just something with one unpaired electron. That lonely electron makes it really unstable, very reactive. It wants to grab an electron from somewhere, anywhere to pair up. And oxygen itself, interestingly, already has two unpaired electrons normally. So it's kind of primed, you could say, to form these even more dangerous radicals if things go a bit wrong. We group these under the umbrella term reactive oxygen species, or ROS. So these ROS, they're either the radicals themselves or things that can easily become radicals. The main ones derived from oxygen reduction are, first, superoxide, O2 with one extra electron, still a radical. Then, if superoxide grabs another electron, you get hydrogen peroxide, H2O2. Now, H2O2 itself isn't technically a radical, but believe me, it's dangerous. And finally, if hydrogen peroxide gets reduced further, you get the hydroxyl radical. No H, this one. This is the really bad actor, extremely toxic, probably the most damaging of the bunch.
Wow. And, it's amazing to think our whole existence hinges on something called spin restriction. You mentioned oxygen's electron setup. Well, the electrons in our stable molecules, DNA, proteins, lipids, they have opposite spins. And this creates a kind of natural barrier. Makes it really hard for oxygen to just react instantly with everything. Without that subtle atomic detail, that spin restriction, life like ours couldn't exist. We'd basically just spontaneously oxidize in the air. It's an incredible safeguard we rarely think about.
It absolutely is. That spin restriction means oxygen is pretty slow to react unless there's a catalyst, something to help it along. Ideally, in our cells, oxygen accepts 4 electrons nice and safely, becoming water. H2O, no problem. But, like you said, the issue is when it accepts those electrons one by one, that's how you get those ROS superoxide, hydrogen peroxide, hydroxyl radical popping up as unwanted side products.
Okay, so if these things are so bad, how do they actually get made in our cells? Is it always just an accident? Or does the body sometimes make them deliberately?
It's actually both. Let's look at the sources. First, there's non-enzymatic generation. A big source of, a constant low-level leak is in the mitochondria, specifically around coenzyme Q, or CoQ. It's key for energy, but its radical form can sometimes accidentally pass an electron to oxygen, making superoxide. Just a byproduct of breathing, essentially. Also, you've got free transition metals, things like iron, Fe2 plus air, or copper Q plus air. They're trouble. They can drive the Fenton reaction, which takes hydrogen peroxide and turns it into that super toxic hydroxyl radical. This is so dangerous that cells are really careful with these metals. They lock iron up in proteins like ferritin, for instance, keep it contained.
Right, so accidental leaks. But what about the body's own enzymes? Do they contribute?
They do. Enzymatic generation is another big source.
Like the cytochrome P450 enzymes. I know they're involved in detox.
Exactly. They're major players, but the way they work, transferring single electrons, makes them prone to, let's say, leaking free radical intermediates. Take carbon tetrachloride, a solvent. P450 tries to break it down, but it actually turns it into a highly reactive radical, CCl3. This radical then escapes and starts chain reactions in cell membranes, causing terrible liver damage. We saw this tragically in industrial exposures. And there are other oxidases, too. Monoamine oxidase, for instance. It breaks down neurotransmitters like dopamine, but it produces hydrogen peroxide doing it. That's relevant for Parkinson's, as we'll see. Then you have xanthine oxidase, involved in breaking down purines. It can make superoxide or hydrogen peroxide, and it's a really big factor in ischemia reperfusion injury. That's the damage when blood flow returns after being cut off.
And, okay, this is surprising. Sometimes our bodies make these radicals on purpose.
Yes. Our immune cells, like neutrophils and macrophages, deliberately churn out toxic oxygen radicals like superoxide during inflammation. It's a weapon, basically, used to kill bacteria or tumor cells.
A powerful weapon, but sounds like it could cause friendly fire.
Exactly. There's always that risk of collateral damage to nearby healthy tissue. And don't forget external factors: ionizing radiation, cosmic rays, radioactive materials, even medical X-rays. They have enough energy to literally split water molecules inside you. That instantly creates hydroxyl and hydrogen radicals. Very damaging, especially to DNA. Can lead to mutations, cell death.
Okay, and then there's another whole category. Reactive nitrogen-oxygen species, RNOS. These come from nitric oxide. NO. NO itself is fascinating, right? It's free radical, but it seems to wear two hats, sometimes good, sometimes bad.
Precisely. At low levels, controlled levels, nitric oxide is absolutely vital. It's a signaling molecule. Acts as a neurotransmitter, a hormone. Famously causes vasodilation, makes blood vessels relax and widen. That's why nitroglycerin helps angina. It produces NO, opens up those vessels.
Yeah.
But at high concentrations, NO becomes a problem. It can react with oxygen or superoxide to form really toxic RNOS, things like peroxynitrite, ONO, and nitrogen dioxide, NO2. Immune cells generate these high toxic levels of NO on purpose, again, to kill invaders, but that's when you get significant RNOS production. And NO's toxicity works in two ways. There are direct effects. It can bind to iron in proteins, like in the electron transport chain, and mess up their function. Then you have the indirect effects from the RNOS it generates. Peroxynitrite, for example, it's a strong oxidant, pretty stable, so it can drift around the cell, causing trouble. It oxidizes lipids, proteins, DNA. It can break there into other damaging radicals, too. So RNOS cause widespread issues: enzyme inhibition, mitochondrial damage, energy depletion, DNA breaks. The works.
Okay, so it's crystal clear these radicals are bad news. Once they're loose, what could they actually attack? Is it just random chaos, or are some parts of the cell more vulnerable?
Well, they are highly reactive, so they're not too picky. But yeah, certain things are definitely prime targets. They basically rip electrons away from vital molecules. Lipids in membranes, proteins doing jobs, carbohydrates, and of course DNA, that causes major cellular dysfunction. Let's start with damage to lipids in membranes. Think about your cell membranes. A hydroxyl radical can kick off a nasty chain reaction. It grabs a hydrogen atom from a polyunsaturated fatty acid common in membranes. This creates lipid peroxyradicals, then lipid peroxides. It's like the fats in your membranes are going rancid, oxidizing from within. These damaged lipids break down into other things, like malondialdehyde. We can actually measure that in blood or urine. It's a marker for this kind of damage.
Huh. And, you sometimes see those liver spots on older people. Is that related?
It often is, yes. That's frequently linked to lipofusin granules accumulating. Lipofusin is basically cellular garbage, cross-linked, damaged bits of lipids and proteins, often from oxidized organelles that the cell can't get rid of. In Parkinson's disease, you see these show up as Lewy bodies in the neurons that are dying off.
Wow. So it's not just cosmetic aging. It's a sign of real membrane disruption. Affects how cells work and could even generate more radicals.
Exactly. Now, think about damage to proteins and peptides. Certain amino acids, the building blocks, are more vulnerable to attack, especially by hydroxyl radicals. Proline, histidine, arginine, cysteine, methionine. The results: proteins can get chopped up, fragmented, or amino acids can cross-link, forming clumps, aggregates that gum up the works. The cell can't easily recycle these damaged proteins. You see evidence of this in cataracts, for instance. The proteins in the eye lens show clear signs of free radical damage, becoming cloudy. Even glutathione, which we'll discuss as a key defender, can get oxidized itself. Its protective sulfur group gets hit, reducing its ability to neutralize other threats.
And the big one: damage to DNA. Oxygen radicals are a major source of DNA damage, aren't they? Leading to mutations, potentially cancer.
A huge source, yes. And what's particularly insidious is how iron, F2 plus RDOG, can bind loosely to DNA. This basically creates a little hot spot. It helps generate hydroxyl radicals right on the DNA molecule, maximum damage potential. This can directly alter the DNA bases, like turning guanine into something called 8-hydroxyguanine, or it can even break the sugar phosphate backbone of the DNA strand itself.
Scary stuff. Does the cell have any way to fix this?
Oh yes, thankfully. Cells have very sophisticated DNA repair systems. They're constantly scanning and fixing damage. But if the damage is just too overwhelming, the cell can trigger apoptosis programmed cell death. sacrifice itself to prevent passing on dangerous mutations.
Okay, so it's a constant battle, but thankfully our cells aren't just passive victims. We have this incredible layered defense system, a whole internal army fighting back against this oxygen toxicity, right? Trying to keep that balance.
Absolutely. The first line of defense is our antioxidant defense enzymes. Really powerful stuff. The star player here is superoxide dismutase, or SOD. Its job is to take two superoxide radicals, the most common initial radical, and convert them into hydrogen peroxide and regular oxygen. It essentially disarms superoxide very efficiently. We have different versions or isoforms. One works in the main cell fluid, the cytosol, and outside the cell. Another crucial one is in the mitochondria, right, where a lot of superoxide gets made.
And mutations in SOD are linked to ALS, amyotrophic lateral sclerosis.
That's right. Mutations in the gene for the cytosolic form, SOD1, are linked to familial ALS. It really underscores how vital this enzyme is.
Okay, so SOD makes hydrogen peroxide, but we said that's dangerous too. What deals with that?
Good question. That's where catalase steps in. Catalase is incredibly efficient at taking hydrogen peroxide and breaking it down into harmless water and oxygen. It works mainly in peroxisomes, these little sacs inside the cell. Its key role is preventing that hydrogen peroxide from meeting free iron and forming the hydroxyl radical via the Fenton reaction. Then there's the glutathione peroxidase family. These are interesting enzymes. They contain selenium. They use a small molecule, glutathione, GSH, as a helper to reduce hydrogen peroxide to water. They can also neutralize lipid peroxides, turning them into harmless alcohols. They handle H2O2 produced outside peroxisomes. And this whole glutathione system needs recycling. That's the job of glutathione reductase. It takes the used oxidized glutathione, GSHG, and converts it back to the active reduced form, GSH. And this recycling step absolutely requires NADPH. You might remember NADPH from the pentose phosphate pathway. It's essential for protecting against free radical damage.
Right, so a whole team of enzymes working together. But what about non-enzyme defenses? Things we might get from diet. Non-enzymatic antioxidants.
Yes, these are often called free radical scavengers. Their basic strategy is to donate a hydrogen atom to a free radical, stabilizing it, turning it into a non-radical. The antioxidant gets oxidized itself in the process, but they're often regenerated.
Like vitamin E alpha tocopherol.
Exactly. Vitamin E is lipid-soluble, so it hangs out in cell membranes. Perfect place to intercept lipid peroxy radicals and stop those chain reactions. When vitamin E donates an electron, it becomes a radical, but it's a relatively stable one. It doesn't immediately start causing more trouble.
But you hear mixed things about supplements, right? Does just taking vitamin E pills work as well as getting it from food?
That's a really important point the source material touches on. While dietary vitamin E seems beneficial, studies on high-dose supplements, especially in people who are already well-nourished, have been disappointing. Sometimes no effect, sometimes even a hint of harm. It suggests that antioxidants work best as part of a complex network, like you find in whole foods, rather than in isolation. The synergy matters.
Interesting. What else is in that network?
Well, there's ascorbic acid, vitamin C. It's water-soluble, and one of its key roles is actually regenerating vitamin E. It can donate an electron back to the used vitamin E radical, restoring it so it can protect membranes again. Teamwork. Then you have carotenoids, beta-carotene, lutein, zeaxanthin. These pigments are good at dealing with singlet oxygen, another reactive oxygen form. Lutein and zeaxanthin are fascinating. They concentrate in the macula of your eye.
Protecting against macular degeneration.
That's the idea. They absorb damaging blue light and quench singlet oxygen right there. So eating your spinach and kale rich in lutein might literally help save your sight.
Good motivation. And flavonoids, you hear about those in tea, chocolate, red wine?
Yep. Flavonoids are plant compounds with multiple tricks. Some inhibit enzymes that produce superoxide. Others grab onto metals like iron and copper, chelating them so they can't participate in the Fenton reaction. And many can directly scavenge radicals themselves. And our bodies make some antioxidants too, endogenous ones, uric acid, for example. It's actually a major radical trapper in our blood plasma and saliva, especially important in the lungs, where other antioxidants might be lower.
And melatonin, the sleep hormone.
That's right. Melatonin does double duty. It regulates circadian rhythms, but it's also a potent free radical scavenger. It readily donates electrons to neutralize radicals, and it can mop up both ROS and RNOS. Plus, it easily crosses cell membranes and the blood-brain barrier, so it can protect many different tissues, including the brain. And beyond specific molecules, cells use broader strategies. Cellular compartmentalization, keeping ROS production contained, often packing those areas like peroxisomes with lots of antioxidant enzymes. Also, metal sequestration, actively binding up iron and copper so they can't cause trouble.
And, of course, constant repair processes. Fixing DNA, swapping out damaged fatty acids and membranes, degrading and replacing damaged proteins. It's non-stop maintenance. Okay, this biochemistry is fascinating, but let's connect it back. Where do we see the real clinical impact of all this free radical stuff going wrong? Can we look at some examples?
Absolutely. The source provides some powerful ones. Let's take Les G and Parkinson's disease. Les was 62, came in with that classic resting tremor, slow movement, stiffness, balance problems, textbook Parkinson's. Biochemically, what's happening is the loss of specific dopamine-producing neurons in the brain, in the substantia nigra. Less dopamine gets to the new striatum, causing the movement issues, and free radicals are heavily implicated. In those dying neurons, we see signs of mitochondrial problems, dopamine breakdown producing H2O2, lower levels of the antioxidant glutathione, and more iron hanging around. Plus, you see those lipofuscin clumps, the Lewy bodies accumulating. It's a whole picture of oxidative stress. There's even a toxin model using MPP+, which inhibits mitochondrial complex I, causing superoxide production and mimicking Parkinson's symptoms. Treatments often target this, like MAOB inhibitors early on to reduce H2O2 from dopamine breakdown. Later, LDOPA replaces the lost dopamine.
Such a clear link there. What about Core N, an ischemia reperfusion injury? She had chest pain, got a clotbuster drug, TPA, but then had a dangerous heart rhythm during the treatment.
Right, that's the paradox of reperfusion injury. Ischemia means lack of oxygen. Cells are starved, ATP drops, the electron transport chain gets backed up full of electrons. Then you restore blood flow with TPA, oxygen floods back in, and suddenly that oxygen rapidly accepts all those backed up electrons. This causes a massive burst of superoxide, mainly from CoQ and maybe xanthine oxidase. This initial burst triggers further damage, leading to hydroxyl radicals. Immune cells rushing in can add to it, making NO and superoxide, forming RNOS.
So the very act of saving the tissue by restoring oxygen ironically causes a second wave of injury from free radicals.
Precisely. That burst of radicals upon reperfusion can stun the heart muscle, cause arrhythmias like chorus ventricular tachycardia, and ultimately kill cells that might have otherwise survived the initial ischemia. Finding ways to block this reperfusion injury is a huge focus in cardiology.
Wow. Okay, and one last really surprising connection you mentioned earlier, TCA cycle enzymes and cancer. How does the cell's basic energy cycle link to cancer through radicals or related mechanisms?
It's not directly radicals here, but it's a fascinating downstream effect related to cellular metabolism and signaling uncovered relatively recently through genome sequencing. They found mutations in specific TCA cycle enzymes in certain cancers. For example, mutations in succinate dehydrogenase cause succinate to build up in some rare tumors. Fumarase mutations cause fumarate buildup in others, like uterine fibroids. And maybe the most studied are mutations in isocitrate dehydrogenase, IDH1 and IDH2, found in brain tumors, like gliomas and certain leukemias. These mutated IDH enzymes gain a new abnormal function. Instead of making alpha-ketoglutarate, they make a molecule called 2-hydroxyglutarate, 2-HG.
Okay, so these metabolites, succinate, fumarate, 2-HG, start piling up. What do?
They act as inhibitors. They block a whole class of important enzymes that normally use alpha-ketoglutarate. These are the alpha-ketoglutarate-dependent hydroxylases. These hydroxylases do critical jobs, like removing methyl groups from histones and DNA epigenetic modifications.
So blocking them messes with epigenetics, how the genes are read.
Exactly. The buildup of succinate fumarate, or 2-HG, inhibits these hydroxylases. This leads to widespread hypermethylation extramethyl groups stuck on DNA and histones. This altered epigenetic state changes gene expression, often making the cells behave more like immature stem cells, pushing them towards uncontrolled proliferation. It also affects another hydroxylase that targets hypoxia-inducible factor, HIF. Normally, HIF gets hydroxylated and rapidly degraded when oxygen is present. But when that hydroxylation is blocked by these metabolites, HIF sticks around for longer, even with normal oxygen.
And HIF promotes growth, right? Like the cell thinks it's starved for oxygen.
Precisely. So HIF stays active, driving proliferation and other cancer-promoting pathways. But the exciting part is this discovery opens up new treatment possibilities. Drugs are being developed that specifically target the mutated IDH enzymes. In lab studies, these drugs can lower 2-HG levels, reverse the hypermethylation, and actually push the tumor cells to differentiate, to mature, and stop dividing uncontrollably. It's a really promising area.
Incredible how these metabolic pathways connect to so many things. So, wrapping up our deep dive today.
Wow.
We've seen oxygen, totally essential. yet this constant source of reactive species that threaten our cells. But we've also seen the amazing defenses, enzymes, scavengers, repair systems, all working nonstop to keep things in balance.
Yeah, and connecting it all, you see this fundamental biochemical truth, right? It's all a delicate dance. Tiny shifts in these reactive species can have massive consequences for health neurodegeneration, heart attacks, even cancer. It really highlights how complex and interconnected everything is, right down to how you use the oxygen you breathe.
Absolutely. So maybe the next time you take a breath, just think about it.
Yeah.
That simple act fuels this incredibly complex, constant biochemical battle inside every single one of your cells. And it makes you wonder, what else can we learn about supporting our bodies in this ongoing fight against, well, the inevitable process of oxidation? Something to ponder.